Hello and welcome back. So we're going to watch a video on photosynthesis, on the photosynthetic electron transport chain. And you can find this actual lecture or this video on the website that I'm going to go to right now. It's linked to this PowerPoint. But if for some reason the sound doesn't work, you can actually just go to the website itself and look at these. It's a free website. It's just a student companion website. So this is on the electron transport chain. I wish I could make this bigger, but I don't know how, so I apologize.
In plants, photosynthesis occurs in specialized organelles called chloroplasts. The internal membranes of chloroplasts are organized into sacs called thylakoids. Surrounding the thylakoid membrane system is a semi-liquid substance called stroma. Photosynthetic pigments are clustered together to form photosystems. When a photon of light strikes the reaction center of photosystem 2, it excites an electron. Two water molecules bind to an enzyme at the reaction center. This enzyme splits the water and uses the electrons from the water to replace the electrons removed from the reaction center. Oxygen is produced in this process. The primary electron acceptor for the light-energized electrons leaving photosystem 2 is plastoquinone. The reduced plastoquinone passes the excited electrons to a proton pump embedded in the membrane called the B6F complex. Arrival of the energetic electrons causes the B6F complex to pump protons from the stroma into the thylakoid space, thereby generating a proton gradient across the membrane. Because the thylakoid membrane is impermeable to protons, the protons in the stroma must pass through the channels provided by ATP synthase. As protons pass through, ADP is phosphorylated to ATP and released into the stroma. This process for making ATP is referred to as photophosphorylation. When photosystem 1 absorbs a photon of light, its reaction center passes high-energy electrons to ferredoxin. The enzyme NADP reductase then transfers the electrons to NADP to form NADPH. Electrons lost from photosystem 1 are replaced by electrons generated from photosystem 2. A small protein called plastocyanin, PC, then carries the electrons from the B6F complex to photosystem 1.
Alrighty. So I'm going to go back to our lecture. I think it's right there. There we go. And now we can talk about cyclic photophosphorylation, which only uses photosystem 1. So photosystem 1 also takes in solar energy, and it will activate an electron. But if we're only using cyclic electron pathway, then we're not going to produce any NADPH. We're only going to produce ATP. So what happens is that electron, when it gets excited, it's going to jump into that electron transport chain. So it'll be picked up by that initial electron carrier protein, plastoquinone, and it will move along the electron chain, transport chain, and ATP will be produced, but the electron will actually move right back into photosystem 1, and then it will move back up in energy levels and jump back over to that electron transport chain. So it's just a cyclic pathway, which only produces ATP. So this is really important in organisms that need a lot of extra ATP for, apologies for my dog, ATP for photosynthesis. So think about organisms that are anaerobic, so bacteria that don't use oxygen. They need a lot more energy to produce a carbohydrate because it takes a lot more energy to pull apart carbon from something like and I'm just going to make this up, but if you are or trying to pull or add carbon, I won't say add, pull carbon, or I shouldn't say that, get the hydrogens and electrons from, so it's not about carbon, it's about getting the hydrogens and electrons from something like sulfuric acid. It's harder to pull those apart than it is to split water. And so you need a lot more energy. And it's also going to take more energy to make up your carbohydrate because you don't have as much energy without oxygen. Okay? Think of anaerobic fermentation versus aerobic respiration from last chapter. That's an example, okay? And so here you can see that cyclic pathway. Again, we just have photosystem 1, takes in sunlight energy, activates that electron. The electron moves up in energy levels, jumps, goes down that electron transport chain. ATP will be produced, but the electron will move right back into photosystem 1. So in general, electrons that go into photosystem 1 come from the splitting of water in photosystem 2. So in this case, we have to keep reusing that same electron. And that's where that cyclic pathway comes from and why we only produce ATP. So the ATP can be used then in the Calvin cycle. And here's what the electron pathway and the photosystems look like within, because I showed you initially, I showed you just that non-cyclic and cyclic pathway. So here you can see photosystem 2 embedded within the thylakoid membrane. Here you can see that initial plastoquinone. Here is the electron transport chain. Here, so this whole thing would be the electron transport chain. Here is photosystem 1. The electron moves into it. Typically what happens is from photosystem 1, the electrons are going to jump and attach to NADP by NADP reductase. Which produces NADPH. And here's that ATP synthase molecule. So when the hydrogens move across, they're going to have to flow back out through the ATP synthase complex, all embedded within this plasma membrane of the thylakoid. So this isn't working, which is why I wanted to make sure I had this. So this is cyclic and non-cyclic photophosphorylation. Same website. And it has the words so you can read them as well.
Photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes down through an electron transport system and finally back to the reaction center. The energy released during this electron transport is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation. No reducing power needed for biosynthesis is generated in this process. Plants and cyanobacteria utilize two photosystems which work sequentially to produce both energy and reducing power. First, a photon of light ejects a high-energy electron from photosystem 2. The electron lost from photosystem 2 does not return to photosystem 2, but is replaced by an electron generated from the enzymatic splitting of water and the release of oxygen. The electron then travels from the excited reaction center of photosystem 2 down an electron transport chain, and finally to the reaction center of photosystem 1. This electron transport system generates a proton motive force that is used to produce ATP. Since the excited electron does not return to photosystem 2, this mechanism for making ATP is called non-cyclic photophosphorylation. When photosystem 1 absorbs a photon of light, it ejects a high-energy electron, which is used to drive the formation of reducing power in the form of NADPH. The ejected electron is replaced by an electron from photosystem 2.
All right. So at this point, hopefully you have a really good understanding of that non-cyclic and cyclic pathways. Just pause that so it doesn't cause problems. We'll go back to our lecture. And so now we're going to talk about some terms that I've already talked a little bit about in last chapter as well as in this chapter, oxidation and reduction. So hold on one moment, okay? All right, so I want to talk about oxidation and reduction, just remind you what those terms mean. Oxidation is the loss of electrons, and in biology it also means the loss of hydrogen ions. Reduction is the gain of electrons, and again in biology that also means the gain of hydrogen ions. So I always remember it as Leo the lion says grr, loss of electrons, Leo. The lion says, or is oxidation. So Leo, L-E-O, loss of electrons, oxidation. The lion says grr, gain of electrons is reduction, okay? And I think oil rig is the other one. Oxidation is loss, reduction is gain. So that's another way you can remember these terms. Of course, most people learn these in their chemistry class, so hopefully that's something you will also remember. We want to know these because in our next step, just like in cell respiration, we were oxidizing glucose continuously. In this case, we're going to reduce carbon dioxide to produce a carbohydrate. And so we're doing basically the opposite reaction. So remember in the last chapter we had that glucose molecule and we oxidized it and produced carbon dioxide. We had oxygen and we reduced it to water. In this case, we're going to take the water, we're going to oxidize it to oxygen, and we're going to take the carbon dioxide and reduce it to a carbohydrate, okay? So it's the exact opposite reaction. We reduce the carbon dioxide in the Calvin cycle, which is the light-independent reaction. And so we're going to take carbon dioxide from the atmosphere that moves in through that stomata, and it's going to attach or move into that Calvin cycle, attaching to a 5-carbon molecule called RUBP. RUBP, ribulose 1,5-biphosphate, is a 5-carbon molecule. When it binds to carbon dioxide, it becomes very unstable and it will split really fast. So the first step is carbon fixation when it binds, and then it's going to split immediately into 2 3-carbon molecules, and then we're going to reduce the carbon dioxide. And then in the end, we have to reproduce RUBP. So carbon dioxide attaches to that 5-carbon RUBP molecule. It produces a 6-carbon molecule, but the 6-carbon intermediate molecule is very unstable, and because of that it breaks down very quickly into 3-carbon molecules called phosphoglycerate. And so because of this, so here we have 2 of these 3-carbon phosphoglycerate molecules that are produced. Now we're going to have to reduce each of these 3-carbon molecules or add hydrogens and electrons to them. So ATP is used in this reaction to help facilitate the reduction and to help facilitate the production of the new carbohydrate. So 3-PG, or 3-phosphoglycerate, is going to be converted into glyceraldehyde 3-phosphate. And so it takes 2 steps. So you have this 3-PG, then you have a 1,3-biphosphoglycerate, and then you have this glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is what we want to produce. So glyceraldehyde 3-phosphate can, when you get 2 of them together, can build up a glucose molecule. So when this happens, we end up with these phosphoglycerate molecules. And I'm actually going to go to the next step because this is just telling you exactly what I just mentioned. Glyceraldehyde 3-phosphate, once we have them, we can use that to produce all kinds of molecules. We can use glyceraldehyde 3-phosphate in producing proteins, or in producing fats, or predominantly producing carbohydrates. And carbohydrates can then be used structurally for storage or as energy sources. But in this process, we have to regenerate RUBP. RUBP is a 5-carbon molecule. So every time we add it takes 6 carbon dioxide molecules, so 6 turns of the Kelvin cycle to produce 1 glucose molecule. And the reason for that is most of the glyceraldehyde 3-phosphate is going to, not 6 turns, I'm sorry, 6 glyceraldehyde 3-phosphates to produce 1 glucose. Because the majority of the glyceraldehyde 3-phosphate that is produced has to be used in regenerating the RUBP. And so for every carbon dioxide that comes in, we produce 2 glyceraldehyde 3-phosphates. So once we have run the cycle 3 times, we end up with 6 glyceraldehyde 3-phosphates. 5 of those glyceraldehyde 3-phosphates have to be used to reproduce the RUBP. So we only gain 1 glyceraldehyde 3-phosphate, which means it takes 6 turns to make 1 glucose molecule. Every time the Kelvin cycle runs, we only gain 1 carbohydrate or 1 carbon of that glucose molecule in a sense. And we need 2 glyceraldehyde 3-phosphates to bind together to produce that glucose. And if this doesn't make sense, I can draw it on the board and show you what I'm talking about. Here's another picture. So this is showing 3 carbon dioxides coming in. So we now have 3 6-carbon intermediate, or 6-carbon unstable intermediates that are going to break down into 6 3-carbon phosphoglycerate molecules, which then are going to be used or be reduced to 6 3-carbon glyceraldehyde 3-phosphates, but we only get to gain 1 of those glyceraldehyde 3-phosphates. The other 5 are going to be used to regenerate RUBP. And I have another video from that same website showing the Calvin cycle.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
So it has to turn 6 times technically, okay? All right. So there are alternate pathways for photosynthesis. So what we've learned about and what I've just taught you is C3 photosynthesis. This is the typical photosynthesis that we think about. Occurs in the Midwest, occurs, well, throughout the U.S. This is what is used in plants like wheat and rice, so a lot of our grain plants. A lot of our flowering plants use this type of photosynthesis. And so we have this mesophyll tissue in our plant, in our leafy tissue, in our leaves, and within there we have the chloroplast, which contains the thylakoid as well as the stroma where we have the Calvin cycle. The problem with C3 photosynthesis is that in periods where there's a drought, the stomata close, and when the stomata close, oxygen that builds up within the plant tissue ends up being used. So RUBP carboxylase will bind to both carbon dioxide and oxygen very readily. And because of this, we end up with this process called photorespiration, where RUBP binds to oxygen and does not produce anything. It goes through that Calvin cycle, but nothing happens. And because of that, it's a waste of energy. And so some plants have evolved to photosynthesize either in different regions of the cell or at different times of day. And so with C4 photosynthesis, the plant actually uses a different region, a different part of the cell for the Calvin cycle. And during the, so the stomata is still open, carbon dioxide is taken in, but the carbon dioxide is going to bind to a 3-carbon molecule, producing a 4-carbon intermediate, which is where you get that C4 photosynthesis. Then that molecule has to move out to the bundle sheath where the Calvin cycle occurs. So this doesn't seem very effective because it takes more energy, but it's actually very good in regions where the weather is a lot drier. So think of Hawaii, where it's very hot, and you have, you know, I guess drier climate, drier climate. So sugarcane is one of the plants that is produced using C4 photosynthesis, and this protects the plant from photorespiration. Another plant that uses C4 photosynthesis is corn. And so that's actually occurs here in the Midwest. But when I think about it, I always think of hot, dry areas. And then we have CAM photosynthesis. So CAM photosynthesis is photosynthesis occurring where it's really hot and really dry all the time. So places that have to really conserve water, like the desert. And so in this environment, we take in the carbon dioxide at night when the weather is cooler, so water can't escape, water vapor can't escape. So the stomata only open at night, and so the light independent reactions occur during the day, and the light dependent reactions pretty much occur at night. So the plant still takes in sunlight energy so that it can activate and the electrons, but the stomata will only open at night. So at nighttime, carbon dioxide comes in, and that carbon dioxide is waiting to move into the Kelvin cycle, but then and it'll just sit there, and then the stomata close during the day, and during the day the light comes in, sunlight energy, and we produce that, or we get the sunlight energy, the electrons get excited, and then we can use the splitting of water to to get those electrons and activate them at night, and that carbon dioxide can move into the Kelvin cycle, and during the day we can actually run the Kelvin cycle while the sunlight energy is being taken in, so we absorb a lot of energy in those chlorophyll molecules, and then they can do their job during the night. The last thing I wanted to mention is photosynthesis versus cell respiration. Hey! Sorry. Plants and animals both carry out cell respiration. So plants and animals have mitochondria, but only plants photosynthesize, so plants also have the chloroplast. Both are going to use that electron transport chain to produce some ATP. The reactions are actually basically the opposite of each other. So the energy produced in cell respiration in plants can be used to drive photosynthesis, whereas the energy that is used in animals that is produced in animals is going to be used to allow us to find food that we can take in, so we get our carbohydrates. And that is the end of my lecture. So here you see the chloroplast and the mitochondria and how they are related to each other. And if you remember, I think we talked about it in chapter 4 when I was talking about different organelles. In that lecture, I had both the mitochondria and the chloroplast are part of the, I can't remember the name. endosymbiotic theory. So very interesting. I'll stop here and I will talk to you guys in class. Have a great rest of your day. Bye. There we go, and stop.